Let's get started. I was told there's spring fair outside. That might be more attractive to some students, but for those of you who are here, you are definitely more attracted by SBE too. That might be overstatement. But anyway, so we'll continue to cover. I will continue to cover for next 2 or maybe 3 lectures is the auditory system I started earlier, and we're coming to the end to a semester. So this will be one of the last block of lectures, and then we'll have one more guest lecture to cover. Now, before I start, I will say that everything we talk about in the class on the slides, you are expected to understand. Not necessarily memorize every word, but you really need to understand the context as a message. And if you don't, and you should grab the TA, go to review sessions. And in the final exam, which we are prepared to put together now, we'll cover the last 1/3 of lectures, as well as all the lectures throughout the entire class. There's a percentage of questions, there's a portion of questions in the final exam that are meant to integrate your knowledge through entire classes. Now, don't get scared. This does not mean you have to go back to memorize every page from day 1, but rather you really should get a good understanding of the important principles that we talked about earlier in the class and emphasize again and again. And also being able to derive from the knowledge you have learned to discuss questions that run across the 3/3 of the class. Even though we give these lectures in blocks, but we meant to have them integrated together to give you some view of the nervous system as a brain. And for example, and when after we've gone through all the classes, I give a lecture on auditory system and somatosensory system. Other professors talk about visual system, and so forth, so on. But if you ask a question that concerning the comparison between all the sensory system, you should be able to answer that. If you ask a question concerning integration of a sensory and motor system, you should be able to derive that question on the spot, because all the pieces have been given to you. So there will be questions in the final exam that are more comprehensive than filling blank, right? And they should expect that, because this is an integrated system class that we don't train you to just fill in blanks. So, but if you really understand what has been taught, it should be fine. But this is my message to all of you, and including those who will be listening to my recording, hopefully, that the final exam will contain comprehensive questions running across all the exams. That has been the case always. I think this helps you to learn this better. Now, I realized that when I gave my last lecture a couple of weeks ago, I ran out of time and I rushed through last few slides and I didn't cover it. So I'm going to go back to finish this part, then continue to talk about the slides I give out handout for today. And now, the last time we talked about these couple of questions, and the last question I try to finish is how is the speech sound represented by neuron firing? Now, we'll still talk about a peripheral auditory system, that is, things happen right after the ear. And after this, and we'll begin to discuss central auditory system. Now, just to refresh your memory, this is a picture that you've seen more than once now. It's very important, very simple, but capture a lot of messages. This is a picture showing the time waveform and spectrum of an English vowel, very standard. This is, if for all the auditory slides, if you remember 2 or 3 of them, this will be one you should remember, because it tells you a lot of things that will go back again and again. Now, what does it tell you? It tells you that if you pronounce this vowel, right, it's periodic. You have a signal like this, and you will notice the periodicity here, and that's because something we talk about here, produced by pitch. For any human being, male and female, you have vibration frequency here. We call it pitch. That gives you this periodicity. Now, if you do spectral analysis, fast Fourier transform, this is what you got. And this is a discrete Fourier transform because we talked about last time, because the signal is periodic. This is from a signal system. And then therefore, you should know that this component is a multiple of a fundamental frequency here in 100-some hertz. That is 1 over the period here. So this comes from periodicity here, and this one is twice, this one is 3 times, so on. So all the component here, we call it harmonics, are multiple integers of the fundamental frequency. And therefore, you can deduce from here the interval between neighboring harmonics equivalent to the fundamental. This is what speech is. This is not an illustration, this is real data. If you take your voice, you say, record from a computer, do FFT, this is what you got. Okay? Now, another feature about here is that a vowel like has, in a spectrum, has an envelope. And it has peaks here, 3 peaks here, we call it formants. Those define what are, whether a vowel is or R or E. Okay? So these are important features of a simplest sound in speech. And the question we like to go through, I would like to take you through, is to understand how are these features represented by the auditory system and eventually by the brain inside, so that we can understand this. Now, the important thing for formant is that if you plot the 2 formants, first 2 formants, F1 versus F2, like here, all the English vowels can be seen as clusters. Okay, is here, and other vowels are here. Okay, so this is based for us when we listen to a vowel, whether we can tell it's or R. It's based on this kind of a clustering formats. Okay, so therefore one can argue formats are important for auditory system to decode. And this experiment that researched in this department, this is by 2 professors who are both retired, Murray Sachs and Eric Young. Both of them have taught about this class, SBE2, or it's a formal class called Face Fund for 4 years, for decades. And Dr. Young retired last year. And there were auditory researchers that did this work in the 70s. I think before you guys were born. Now I realize how young you are. In the 70s. And this work showed that if you present vowels, like what we saw, English vowels, to a cat, and record it from nerve of cats. A cat's nerve in here is actually quite similar to other larger mammals, primates, and humans. And then you reconstruct the firing to see whether cat's nerve can recover those peaks called formants. These are 3 spectrum of the vowels E, R that they use. You see, you can see formants location slightly different. So what they did in the experiment is the following, as I show here, and done in 1977. So they presented this vowel many times. You know, this is E. You play E, 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 E. You know, 100 times, 200 times, 100 times. You see. Okay. Now, given what we discussed earlier in class, then you know that every time you present a vowel, there's a neural response. There are spikes. Then you know how to calculate what we say average firing rate. Right? You know all of that. You should be able to do that by hand by now. So this is what they did. This is why this is not trivial. It's simple but useful. So once they collect that many times, they calculate mean firing rate. A plot here. They call it total rate. Means the auditory fibers, as all the neurons, have a spontaneous firing. If you do nothing, the neuron will have firing. That's spontaneous firing. Then you play some. There's called a driven firing. Together, you call it total. So, but this is mean firing rate in that sense. If they record from 100 of neurons, each of them has a frequency tuning. That's called a characteristic frequency. It's a part here. So each dot comes from 1 nerve. They did an experiment. They record from a nerve, play the valves 100 times, calculate firing rate, put 1 point here, another point, another point. Okay. So this experiment runs for hours and days. It's a very gruesome experiment that you have to do this continuously until you collect all the data. So what I'm showing for here is that this is total rate, but if for every neuron it has a spontaneous firing rate, the spontaneous firing rate is different from every neuron. If you take that out, then what is left is called driven rate. That is a change of firing rate caused by By what? By this stimuli. Okay. However, there's one more property you have to take into account. That is, every neuron has a maximum firing rate. So I think I have slides showing here. So let me jump here and go back. So if you take a neuron, auditory neuron, play a sound at a particular frequency, increase sound level, and the firing rate would keep increasing until some point it saturates. Okay. Now, some neurons have firing rate as high as here, some as low as here. So each neuron could have its own maximum firing rate. So that's if you turn the knob, make sound really loud, as long as you can deliver without hurt anyone. The neuron could fire 100 spikes per second maximum, or 200 spikes per second maximum. Okay. So in order to account for that, they normalize this. So after they take the total rate minus spontaneous rate, then divide it by the maximum firing rate a neuron can give. Then it's called normalized rate. Now, here is unitless number. It's no longer spontaneous spikes per second now. Okay, and that if you smooth this, that is, you use the window to smooth data point, then what you can see from here, this profile really is similar to this profile in the sense the first format is here, and the second format is here. But third format and second format is very close. In this case, you cannot tell. Okay, so this is one of the first data that scientists gained that indicated that is an important feature in speech sound like a vowel format can be captured by population of neural firing in auditory nerve. Okay, now in retrospect, people may say, well, it has to be this case. Otherwise, how would we hear it? But you have to show this. Until you show, people actually have no idea whether this can capture it. It's just like, take an example, I say, take my hand, put on the Xerox machine, push a button, my hand come out. You everything take this for granted. But when first Xerox machine was produced, pretty crappy. Put my hand there, come out, maybe just a blob, right? So this is what it shows that auditory nerve as a population have the ability to represent those. Okay? Yes?
Below each horizontal axis, there's a horizontal. There's kind of a horizontal line of dots. So
this one, yeah, I think they're outliers. They're outliers. Yeah, good question. Yes, like this one too. Yeah. Thank you for your question. Yeah, please go ahead. Raise your hand if I didn't much a point. Okay, so let's continue work on. So why is this important? So this is a representation of the vowel spectrum by auditory nerve. Okay, that is a representation of this. Okay, so what about these? What about the temporal structure? Do they play any role here? And now, continue for this, to talk about this. The neurons in auditory nerve, some of them have low spontaneous firing, some have high spontaneous firing. So what you see here is that for no spontaneous firing neurons, the firing will increase and saturate. Okay, saturate. Saturation is a problem. That means if you keep increase the sound level, there's no change of firing. Therefore, neuron will not know what has been changes. Okay. Now, so the problem, the interesting thing for what they show here is that if you present vowels at moderate or low sound level, 34 dB, 30 dB is very faint. It's 34 dB. It's about this. Okay. So when I talk about this now, it's already 60, 70 dB. Okay, so a normal conversation is this range. So what you can see here is, so for this population of neurons, 34 dB, you see two formats, very clear. So they make sound louder, 44 dB, 54 dB, so far so on. As you can see, you can still see the formats here, maybe here, but they're not here now. This thing become flat, now flat. Why is this? Because by the time you hear, you are in the saturation range now. Okay, so that poses a problem. That is, if you see the nerves, auditory nerves use firing rate to represent the vowel spectrum. You can only do so at a low and a moderate sound level. But as you know, when we say right, make 100 dB, you hear is still So you would expect the consistency across sound level for our perception. Okay, so. But then they discover, if you look at the neurons, auditory nerves with low spontaneous firing rate, these neurons have very little spontaneous firing rate, below 20 spikes per second. Even at a pretty high sound level, you can still see those formats. Okay, so altogether, and I skip all details now, is that if you look at all auditory nerve fibers, 100 of them, some of them have high spontaneous fibers. They have a low threshold. So when just turn sound on, while is pretty faint, this fiber began to fire. As you turn the level higher and higher, then high spontaneous nerve fiber becomes saturated. They become flat, they cannot tell anymore. Whereas the low spontaneous fibers have a higher threshold, they begin to fire. So they take over at a high sound level. So together, the vowel spectrum can be represented. This is this message here. And this is related to mean firing rate we talked about earlier. Now, what about the waveform? We say that vowel like also have a waveform modulated periodic, right? And you should probably remember we talked about something called a face locking earlier, right? So that is in the hair cell. In the hair cell, remember when this basal membrane got to push up and down, this hair bundle got to push left and forward and backwards. Only one direction the channel open, therefore depolarize, therefore spikes. The other direction is not open. So as sinusoid continues 360 degrees, only half of the face you have firing, another half no. That is face locking. This is very, very important. That face locking gives auditory nerves the ability to track time change signal, right? Because it only fires every cycle also, okay? Now, because the vowel has time structure, when you present a vowel, then the neuron firing would like this, okay? This is the period histogram. We also talk about how you calculate this. That is, take a firing spectrums, you make them chunk of multiple period, you average them, then you will see this period here. So here are examples of 4 neurons where period histogram was computed using the firing. Okay, this figure has a legend. I suggest you read how they computed this. And for example, this is the same vowel. Why this period is only, if this is the same as the number, right? So you see this period is longer than here. Why is that? Think about this. I take a same vowel, but then there are auditory neurons tuned to 100 hertz, 200, 500, 1,000, 2,000, all of them. You same vowel to present it to them. Then you look at face locking. If you look at neuron at 100 hertz, what will be face locking? What is face locking to, roughly? Versus 1,000 hertz. Right. So here is what a neuron tuned to best frequency. This neuron is tuned to 410 hertz, and this neuron is tuned to 2,220 hertz. Okay. Now you think about this, right? So that's 5 milliseconds. 5 milliseconds is how much? How many hertz? 200 hertz, right? 200 hertz. And so you can figure out actually this periodicity here is directly related to the frequency tuning, because the neuron, remember, now we go back to fundamental about auditory nerve ear now. In the cochlear, there are many hair cells that line up at a different location of a basal membrane. Remember, we talk about each of them serve as a bandpass filter. Bandpass filter at different location frequency. Right? That's the whole cochlear does, is frequency decomposition. You give this spectral wide broad stimuli, then all the nerves hair cells lining up their basic filter. There's a filter like this, right? So the neurons tuned to 400 hertz essentially is bandpass filter at 400 hertz. And when you give a vowel spectrum is pretty wide. You might have overlooked this when I flash it here. Let's go back and look here. This spectrum runs from 100 hertz to about 4,000 hertz. Okay, so therefore, if you have a neuron that tuned to 400 hertz, it sits here, what it got? It got a signal around 400 hertz. Therefore, if it has a face locking, more or less is face locked to 400 hertz. Okay, because earlier when we talk about face locking, we use a pure tone as an example. I give you one pure tone, assume a neuron is tuned to a pure tone. Therefore, face locking is to that pure tone. I change it slightly now. Might be a little confusing, but it's the same thing. That's now I give you a broadband signals. But because a particular neuron is tuned to particular frequency, therefore that face locking we talked about earlier still apply in this case. All right, is that clear? Yeah, this could be a little bit confusing. So that's it. That's the message I want to get here. So anyway, let's continue to do this. So the question is that if a valve has a temporal structure like this, and if every auditory neuron has a phase locking, the question is, can you use phase locking as a way to represent this valve's information? The answer is yes. This is what is analyzed from the same experiment, but different data. This is data based firing rate. Okay, so this is analysis with this formula about phase locking out of each neuron. Okay, so you compute phase locking from spectral of this, and you compute the number from here, which I will let you to read. Then you plot that number that they call average localized synchronized rate. The basis, this is a measure of phase locking in this neuron when the valve is being presented. Okay, not average firing rate now, is the average phase locking amplitude. If you plot that as a function of neurons frequency tuning, you still have a profile like this. Now what you can see here is that this profile shows the format as average firing rate profile. But most importantly is that when you increase the sound level from low to very high, remember firing rate becomes saturated here now. But a phase locking based measure continue to track this all the way to high sound level. So that suggests the auditory nerve might make use of a phase locking to decode the valve at all sound levels. Okay. Whether actually auditory system use phase locking and average firing rate to decode the valve is not an entirely resolved issue. But this show you, if you use this measure, you can get this. Okay. These two examples, the reason I bring up is that average firing rate and phase locking based representation of valve serve as an example for you to understand more deeply what is the basic property we talked about earlier, that is frequency tuning and the phase locking at the aerosol level. And this is a direct consequence of that. Now, I know this part is, it might take some time, so I suggest you to read through these handouts that I have notes on and discuss with your TAs if this is not clear. Okay, that's what I want to finish for last one, slides that I rushed through. And remember, we did not ask any question regarding this in a last exam, but this will be part of the next exam now. Okay. So with that, I'm going to move on to today's lecture and continue from here. That is the central nervous system. So what I know is the following. So so far we've talked about a peripheral auditory system that is from ear to auditory nerve. Okay, we'll talk about how the frequency selectivity is produced here. And then we just spent about 15 minutes or so talk about how the auditory nerve here as a population can represent the valves. Okay, so then the rest of the system is the following. So signal travel from here to the next stage called the cochlear nuclei. And this is where central auditory nervous system begins. So central auditory nervous system contain a lot of neurons, a lot of structures. From here, you go to next stage here. I put a number here indicating synapses after the hair cell. Hair cell will be number's first synapses. Auditory nerve second, third is cochlear nuclei. And then the structure here we'll talk about in a few minutes called medial superior olive or lateral superior olive. Okay, and they do binaural processing. And from there, it continues goes to next stage called the inferior colliculus, thalamus, and auditory cortex. Okay, that whole thing is auditory central auditory system, and we can only cover some snapshots of this because of time. Now, you can imagine there's another ear on this side, so that signal goes through here, goes through pathway and here. So therefore, I should have, we should have a plot on a mirror image here. So all the nuclei from here on receive inputs from both sides, because remember, signal here can cross midline. That's where binaural processing begins. So I mentioned earlier in my lecture, I just reiterate this now. And what makes auditory systems different? Now that you have seen visual system, you have seen somatic system, you go back to take a look. This is a very important thing for you to get an impression on what is the major difference between sensory systems. No sensory system are the same. Auditory system has a longer subcortical pathways that are from here to here. We'll talk about 7 synapses. Okay, and you should have the recollection on how many synapses in the visual system. How many? Anyone still remember from Dr. Connor and the Nielsen's class? What is equivalent to cochlear in the visual system? What is equivalent to the cochlear? Retina. Good. And then after retina, where does signal go? What's the next stage? And what's after retina? What's next stage?
Chiasm, optic chiasm.
Sure.
That's a synapse there where it goes through.
Right. So there is optical fiber. That's a fiber. But where's the end? Anywhere end ends. What's that? And here, someone, I heard a couple. Someone here say something.
Retinal ganglion cell.
Retinal ganglion cell is still within the retina. It's the last stage of the retina. Let's say after that, when what I mean is this, when signal leaves the retina, leaves the ganglion cell, you have action potential now. That goes through optical tract. And the question is where that ends. Where I project to? Anyone? So what's that? Spherical is not quite. You guys remember the power structure called lateral genic nuclei, LGN? That's part of the thalamus. Okay, that's not what we see. Thalamus has a visual part called LGN, and an auditory part called MGN or MGB, and some sensory part called ventral lateral. But anyway, it's a thalamus. So that means if I put a retina here, I would have run a cable direct into the thalamus. That's what a visual system is. Okay, first of all. So now you can see auditory system has all the structure in between. Okay, this is something that is very obvious when you compare two systems. So this is what we mean by longer pathways here. And I explained this before, and I'm going to touch upon from today and the next week, and about the localization and about the speaking vocal production. Okay, so I expect you get a good sense on what does this mean. So this we talk about. We say it's time variant input signals, which we'll talk about what I mean by time variant. Why is it important? And then we'll talk about also the localization issue that is sound into our ear from all the directions. And finally, before I finish all my lectures, we'll talk about this sensory motor integration, that when we listen to something, the sound can come from outside, come from our mouth, or sometimes inside completely. When you silence reading, we will have hallucination. So these are things that we'll touch upon. Okay, but this is sort of a big picture for you to keep in mind. And then we're going to discuss couple examples here. Okay, now here is another view to dissect the auditory nerve to cortex pathways, and don't be bothered by these terminologies. This is pretty awkward ones, but. Don't try to memorize it. I don't even memorize this. But the point here is for you to see, again, from a hair cell, auditory nerve, how this can go. Remember, there's a mirror part here. So there's a part of this ear, there's a plot. So anyway, from here forward, you can imagine it's a binaural. That's a picture, okay? Now, what is important about the central nervous system processing? Yeah? And there are many things going on in the central nervous system. I think there are 2 points, 2 principles that are extremely important for you to understand after all those lectures, mine and others included. That is, central nervous system neurons can extract or compute stimulus information not explicitly encoded by sensory concept, by sensory receptors. Okay, and I'll explain it further with examples, but be sure you understand what does it mean and be able to explain at the end of this. Secondly, central nervous system transform isomorphic or faithful representations of a sensory stimuli established in the peripheral to non-isomorphic representations. So bear with me for what does it mean until we get the lecture. So these are 2 examples, 2 principles I want to illustrate with real experimental data not meaningful. Okay, so auditory system, auditory cortex is actually has some historical association with this university. I think you should know. Now, let me just show this first. Some of you might know, at least from your visual cortex lectures, the name of David Hubel and Thomson Wiesel, right? Because almost every visual cortex lecture textbook have to talk about it, because they are two scientists who know the Nobel Prize for Medicine in 1981 for their discovery of one, the visual V1 structure, orientation, tuning, and columns. Second, the development of those columns, because if you close eyes, as you probably have heard this of a kitten, then the column doesn't exist. Okay, but lots of people didn't know that David Hubel, when he was a young assistant professor, he actually worked here in Hopkins. Okay, Hopkins. He was assistant professor in School of Medicine in the Weimar Institute. And his laboratory, the building where his laboratory is still there today. If any of you had a problem with eyes, go to see a doctor at the Weimar Institute. The basement is where David Hubel's office is. A lot of people don't even, still don't know that the first structure they work on is not a visual cortex. It's auditory cortex. It's what I'm working on now. Okay, so this is a paper that published way before you guys were born in 50s. Where they recorded from a cat's auditory cortex. This is very interesting historical notes, because a lot of people, a lot of research now don't even know these things exist. But it's interesting that they drop an electrode in a cat's auditory cortex, play sound, try to drive this. And here's, back then there was no digital photography, no computer digitized things. So this is a picture shots of the oscilloscope. If you guys are nerds, you should know what a oscilloscope is. So here's the sound they play. There's action potential you saw here. But here's what I saw. Basically, they could not drive this neuron. They say this unit referred to the neuron. This unit almost never responded to clicks, tones, or noise from a nearby loudspeaker. You all know what does it mean. Okay, so they can't drive this. They run out of the idea what to do, because these days, without a digital computer, you cannot synthesize vowels and so forth. So on. They can only play tone, noise, or click from a hardware-based stimulus generator. So, but interestingly, they found that these units respond briskly, for example, to voice squeeze emitted by squeezing a toy rubber mouse, scratching fingernails on the table nearby, hissing, typing on the table. Okay. And the famous story they've told other scientists over years is the following. Many scientists know this story. When they fail to drive a neuron, take out a keychain, go in there, do this, and the neuron fire. Why is this? You're an engineer. Why? What is in this? If I generate this, what sound do I generate? What's in this sound I just generate with this keychain? Is that pure tone? No. It has a multiple frequency. It has multiple modulation. Now we know it's a frequency modulation. That's complex sound. But since they don't have a computer, they cannot control this. And they did all of the tricky stuff. And then they eventually conclude. Finally, they say, this is the end paragraph of the paper, it has proved impossible to discover the stimuli adequate for driving many of our cortical units. A fact we cannot readily explain. They give up that, they move on, work on visual cortex, the rest is a history. So they worked on Weimar for a couple of years. And then the chair of that department took the chair of neurobiology in Harvard. When they become chair, and took along these two junior faculty with them to Harvard, to Boston. So, but what's interesting is David Hubel, and he just passed away a couple of years ago. He's a very highly respected scientist. It's about 8 years ago. I was in the national meeting, Society for Neuroscience meeting. He and Wiesel, both of them just published a book by Oxford Press, recount of their earlier work. So they were sitting there, signing the book. You buy a book, they sign there. I thought it was a wonderful thing to do. So I went there, bought a book. I wanted to say, Dr. Hubel, would you mind signing my book? He said, of course. Then he looked at my, I have a name tag. He said, "Oh, you work in Johns Hopkins?" I said, "Yes." He said, "You know, I used to work there." I said, "I know." I said, "My life just across the street from your old life." Then he said, "What do you work on?" I say, "Oh." So what do you work on? I say, "Auditory cortex." Then he told me one sentence. He says, to many, many people that I heard secondhand until that moment. He said, "If I were to live again, I would work on auditory cortex." He said that for a reason. A lot of people didn't understand why he said that. Because he got a Nobel Prize by working on visual cortex. Why he still think about auditory cortex? Because the very earlier year, he worked on auditory cortex, could not figure out what's going on and move on. But his thought is still on this. Why is this important? Because of the things I told you at the very beginning of my lecture of audition. Why audition is important? Speech, music, and many things. People consider these are some of the most important functions for human beings, above and beyond what an animal can do. Okay, and this is what scientists believe auditory system is an interesting one. Now, many years has passed, and we have done a lot of experiments to some of them can explain why he couldn't find out then. So here's something, a message I want to get to you now is that when we talk about neuron, I jump very far, so bear with me. So when we talk about neuron in auditory nerve, I never said a neuron cannot be driven, right? If you have auditory nerve, you give a tone of a proper frequency, you drive that neuron, right? This is what we see so far. However, if you move through this stage all into auditory system to the cortex, now it's about 5 to 7 synapses away from auditory nerve. That statement is no longer true. That is the following. There are neurons in auditory cortex, such as the one that Dr. Huber encountered in 50s. They are very difficult to drive. Here's the following. You got a neuron, we play sound. Here's the example. One of study done by my former students, we tried all kinds of sound. There are tones, noise, clicks, amplitude modulation, frequency modulation. Here's tone. There's no response. This is spontaneous firing. Okay, until we find something called a 2-pep. This is basically, we give everything we try, almost like what David Huber did earlier, but more sophisticated, because now we have digital computer. We're engineers. We can program MATLAB. We can produce anything we can imagine. You can imagine. Nothing. Until we try this thing called a 2-tone peps. That is, we have 2 tones. One first, 1 second. They differ by slight amount of time, by frequency. It's like boo boo, boo boo. Then we keep changing the distance in time frequency. And this is what we did here for this one. We tried 300 pairs of all combinations. Out of them, only 2 combinations drive it. The rest said no. How long does it take us? An hour, 2 hours, 3 hours, long time. So this is exactly explaining why they could not drive a neuron here, because they didn't have a computer to control the stimulus space back then. And they know this neuron is auditory neuron because they can plot their kitchen and drive this, but they cannot describe this. Now, we describe it now. So in this case, this neuron can be driven by 2 unique combinations of tones. Why is this interesting? Because if you look at speech, when we say ba, ga, this translation has a combination. So when I say vowel, this is very simple, probably simple speech. But if you go anything beyond this, say today, tomorrow, if you look at all the combination, that combination of frequency time, that give rise to complex syllables. Okay, and that's indication that there could be neurons that are selected for this. So if we compare the neurons like this is how selective they are. They almost respond no response to pure tone. There's a firing rate. But if this neuron will give them 2 tone peps together, it responds much stronger than pure tones. And where they are, now you probably have some knowledge from all the lecture here so far about the cortical layer now, right? Cortex has 6 layers. There's upper layer, middle layer, and a deep layer. These neurons that we call the nonlinear neurons, dark dots here, are mostly in the upper layers. This is where human visual in V1 find the most by and large orientation neurons. Those neurons are tonal orientation. Remember, in visual system, if you look from a retina, look at thalamus, LGN, neurons there are not tuned to lines, right? What are tuned to? Center, surround is dot, right? A dot and a surround. Only in the V1 neuron become to tune to orientation. Okay, and the selectivity of neurons like this is reminiscent of that type of response. So now we can go back and see why Dr. Hubel cannot drive this neuron half a century ago, because this neuron are highly nonlinear, highly selective. This is actually a very good example for biomedical engineers, because this is type of experiment. If you're not an engineer, if you don't have capacity to command the computers, and if you don't have an understanding of mathematics and signal processing, you will never be able to go over crosses. If you're a pure biologist, you will not do this kind of experiment. So every graduate student I have in my laboratory are engineers by training. So all of them knows computer, so far so on. So this is actually a very good example to demonstrate that your engineering skills can be very useful to very important and difficult biological problem. Now, the notion we've learned over a year is the following. This is the one summary slide. That is in auditory nerve, and what I illustrated here, the blue color is kind of a schematic of recipient field of a neuron. The red area is within our recipient field. You can drive a neuron very strongly, and we call this a peripheral stimulus. In auditory nerve, there's almost no difference. That's why I said earlier, you get auditory nerve, you play tone, play noise, play voice, whatever you play. As long as the stimulus energy overlap with the receiver field, you drive that neuron. Whereas in cortex, as you go through here, within the receiver field, there's only a very small area where you can drive that neuron. And this is achieved progressively. Okay. Now, some of you ask, why you want to do this? Why can't we just use auditory nerve as a machine for the brain? But the problem with the auditory nerve is that there are 30,000 of them, right? They're all tuned with different frequency. But you can drive them with everything. Therefore, if you speak, I speak, birds singing, car driving by, all of the noise will excite auditory nerve with a particular frequency. How do you put them out? You can't. Whereas by going through multiple stages of processing, eventually, in a higher level of auditory system, neurons is only selective a very small portion of acoustic space. That is, you can imagine, if you look at my neuron in my auditory cortex, that when all of you began to talk, there are male, female, and voice, there are all others and fines that are going, one particular neuron is only listening a subset of the sound, so far sound. That's in principle is how the auditory system is able to extract the signal of noise from the background. Okay. Now, the first experiment I want to tell you today, and I have a few minutes to finish this, is how does the auditory system represent time variant signal? So remember, we emphasize again and again what is unique about auditory system. One of the things that's very unique about auditory system is time dependent. Here was the following. I use the example again. I used it before. So if I put my hand here, your visual system got this image. It got in my hand. I turn around, it got another image from the back of my hand. Remember, either this image or this image has no time component. You can define two-dimensional, three-dimensional in color space, however you want, right? But there's no time. But if I ask you to give me equivalent of this versus this in auditory system, what is the equivalent of signal without time in audition? There's none, because any signal in auditory domain, acoustic signal, has a variable of t, that's time varying, right? So sound, whatever sound, is like a river running on a stream, never stop. Okay, so never stop. That's why when you do Fourier transform, the ideal Fourier transform is infinity, goes back in the future, back in the past. Right? So now, why this is a problem? Because if I give you a sentence to say, today is Friday, it's a long sentence. Today is Friday, it's a long sentence. Now, what's happening in my ear? Auditory nerve. Well, there's action potential that's also go along, today is Friday. Now, so that signal that supposed to represent the signal coming today is Friday, this is S of t, that's the t is the verb time. They say the signal in my auditory nerve that firing along with this is signal they call Y, okay, Y of t. But in both cases, it's the same t. So you have a signal that's time varying. You have its response as time varying. You have two time axes. This one to represent this. How am I going to do this? Well, what an engineer would do is the following. If I say, oh, representing my sentences, today is a Friday. Well, what you do? Well, you turn on your computer. You have digitizer. You have recording devices. Okay? You just turn on. You capture every second, every millisecond, every microsecond of my voice. You say, now I capture it. Your voice is here. Okay? This is what engineers do. This is what a most intuitive thing to do. But the question is, can nervous system do this? Or does the nervous system do this? Answer is no. For many, many reasons that we're going to go through. And here I'll show you, within your ear, within your brain, when you represent my sentence, today is Friday, you don't do this. You don't do what a computer do. And you do something more interesting. So here's that go, okay? So I take that example again. Today is Friday. If you take the sentence, it has multiple components. There is a slow component, slower than 50 hertz. This is called syllable words. Today is Friday. Usually there's only a few hertz, 10 hertz. Okay? And then, but in that same sentence, if you listen more closely, there's something called pitch and periodicity. That is, when I say today is Friday, or a female speaker say today is Friday, you immediately tell us that difference. That is because of pitch. We said many times. That pitch comes from periodicity. That is this kind of, I basically enlarge this. Time scale, remember, it's 100 millisecond, 10 millisecond, 10 times time scale now. Now, that's pitch and gender. Yet more, if you look at even finer detail, and even take two male speakers here. One is me, one is this, your fellow students. Our voice is different. We have individual identity. So when you pick up phone, call your parents, call your friends, you all know that one word, when your parents call you, hi, you did not say, hey, I'm your mom. Just say, hi. You know who is talking, right? That coming from here, we call it fine structure. That tells speaker identity or local dialect. Someone come from Texas, Midwest, California, New Jersey, Florida, all of that carry very subtle difference. So come here. So that means that for our audio system to, when you listen to sentences, today is Friday, you know this, what does it mean, what is gender of speaker, who is talking, if this is someone you know, all of this, instantaneously you know all of this. But you need each of the information at a different time scale. Okay, the time scale. Now, the question is, how does audio system do it? Again, if I say your task as an engineer with the computer you have to capture all of this, how do you design your computer? How do you design your digitizer? I'll give you all the number here. I say, I want you to capture all the information here so I can tell all of this. What you do?
I can't think of anything besides just iterating through all of it.
Well, what it means is how fast you should digitize.
How fast what?
How fast you need to digitize my voice, the sentence I just said, today is a Friday, in order to figure out all of this.
All the way down?
Yes, absolutely. Yeah, you take the finest scale, you digitize that. You say, now I'll capture everything. Because if you capture this, you should capture this, should capture this. Right? That is the principle all of us learn in signal processing. That you take the highest frequency, the fast variation, you digitize to satisfy that twice like with the frequency, then get everything. Okay. Now this is another example of this to say is that if I want to measure distance from that wall to this wall, but at a resolution of 1 millimeter, what kind of ruler are they going to use? Right? Well, you cannot use the ruler this long because it's going to take forever. I say I give you 1 minute, 30 seconds to do this. So then you need a ruler, probably several meters long, but a scale as fine as millimeter to do this. Well, that's exactly the same thing. But that's not how the auditory system do this. Why is that? Because as you know already from signal processing, if you could digitize everything down to 1 millisecond so that I can capture this, then you will waste a lot of things to capture this because you don't need this. Okay. And furthermore, the nervous system simply cannot digitize at this speed at all. That's a fundamental limitation we're going to see here. So here's the basis for all of this. Then we'll talk about, then if the nervous system cannot do this digitization, then what can it do? Because at the end of the day, we know, we listen to that sentence, we know all of this. So all of this has to be represented, right? So this is a very interesting biological and a computational problem. So here's also where everything started that I mentioned earlier. And this is the slide you sent before. This is how the face locking is formed. Okay. Now, if you measure this face locking from all auditory nerve, some tuned to 100 hertz, 1000 hertz, 10,000 hertz, measure quantify face locking. Remember, I discussed this before. You have seen this. Then, for auditory nerve, the upper limit of face locking is this curve. This is data from cats, probably similar to humans. So it's about 1, 2, 3, 4. It's about 4 kilohertz. You completely lose this. Your best up to about 1 kilohertz begin to drop down. Okay, this is what we talk about. Already talk about it. So now let's look at next one. So since auditory nerve sent spikes into next stage called a coccynuclear. So if we repeat this face locking experiment in coccynuclear, what do we got? We got this curve. So this curve moves to the left now, meaning what? Meaning the neurons cannot follow as fast. The upper limit of face locking is below 2 kilohertz now. Why is this? This is a too long story, but since you have taken SCB1, then you actually know this. I mean, that is when the action potential travel from here to here, it has to be integrated. And I don't know if you guys talk about SBU1, and Dr. Winslow talk about this. When the action potential from one neuron to the next neuron arrive at the dendrite, then create a synaptic potential that travel along the dendrite into soma. This process effectively is low pass filtering. So that is you filter the high frequency. So it's inevitable when action potential travels through one synapse another, this becomes slower. Basically, higher frequency get filtered. Now, if we continue to repeat this experiment in inferior cortex, thalamus, and cortex, here's the curve. And schematically, inferior cortex is here, less than 1000 hertz. Thalamus, less than 300 hertz. Auditory cortex, almost only 100 hertz. That means if you do this face locking experiment, you should not expect any face locking faster than 100 hertz in cortex. Does that cause problem? And we said earlier that you need all of this. Now, if you digitize this, put on a CD, do you know what is digitization frequency for CD? Anyone knows this? I think I told you guys earlier, about 40 some, 40, I think 40 some K, right? But anyway, so if I told you that you filter out anything faster than 100 hertz, because there's no more face locking for auditory system, right? Auditory cortex can face lock no more than about 100 hertz. If you take my sentence, today is Friday, or piece of music, whatever, you low pass filter anything above 100 hertz. What do you got? Garbage. You can't understand this, right? So this actually become an issue. We said earlier that face locking is important, but now I'm telling you in the cortex that face locking is really limited by 100 hertz. Okay. So how could a cortical response account for perception now? Okay. So here's the experiment we did a number of years ago. Okay. Using the stimuli, I gave it to you to do your homework. So now you know why I give you that to do homework. This is stimuli you played with earlier in one of my homework. It's collectron. You guys will still remember this, right? So when you listen to this, there's somewhere around here, your perception changes from discrete to continuous. But in this case, we did a following experiment. We got a marmoset. We put an electrode in the cortex, record a neuron. We play here. So we play this to monkey now, right? And we record from neuron. This is what happened. This is one of the neuron that we play the stimuli. For example, this one 10 times. This is dot raster. You all know what is dot raster is now. Good. So 10 times you see face lock very rigorously. This is duration of stimulus. Then we start increasing speed or decreasing the interspec interval. Say face locking pretty good, pretty good, pretty good. Oh, become fuzzy. Nothing. Now, what does it mean is that at this range, it is 30 hertz, 30 milliseconds, or about 30 hertz, all the way to 300 hertz. After initial onset, there's no response. This neuron has no response. But did you hear sound? Could you still hear sound when you did the experiment? Yes. And we're sure that actually these monkeys hear this as well. So this is what puzzled people for a long time, is that phase locking in auditory cortex can only go down. It's about 30, 40 hertz. When I say 100 hertz, that's very generous. It's only about here, or nothing. This is very puzzling because if a neuron in your brain don't respond to sound, you should not hear it. So what is missing? Well, it turns out there are other neurons in the cortex. It's a different neuron now. When we play the same set of stimuli, it did the following. It didn't respond much when in interval was long. As we shorten the interval, that's as you hear speed, click become faster and faster, becomes response more and more. How do we know it's response? We can calculate mean firing rate plot here as a function of this. See, firing rate increase now. So what does that mean is following. That in this range, when sound become very fast, neuron like this cannot capture, and then this neuron capture now, but in a way different from our neuron. Okay, so let me finish one more slide before we go. So we call this isomorphic faithful response because this literally capture physical structure over here. But this is not isomorphic because if I just give you this, you could not tell what's this. However, when this stimulus parameter change, there's rate of fine changes, so it can be encoded. So if we put all the neurons together, the neurons that can follow, synchronize, is this population. This is a vector strings that you can quantify. The neurons cannot follow, but they have a firing rate of here. Two together can capture that whole range. And this is where, when you filled in your homework, your perceptual boundary of discreteness changes here. Okay, so that range you hear and the monkey here is about the same. Okay, so I'm going to stop here, and this requires a bit more understanding. So think about this, and it goes to TA if that's not clear. And I'll continue this Monday.